Good morning, friends. This is a series of lectures related to 2IBC Analytical Chemistry, chapter number 4, Parameters of Instrumental Analysis, semester 5 2019 pattern. Now, in this lecture, we are going to learn about the developing the procedure. Now, the procedure is the important part of the analytical chemistry. In the analytical methods, after once analytical method is decided, it is necessary to develop the procedure. But for what things take into consideration for the for the development of procedure, it is very important. So first point, developing the procedure. In developing the procedure, we have to consider the terms like calibration, standardization, sampling, validation, and protocols. Now, calibrations and standardization, sampling, these are three also important parameters. In this lecture, we are going to learn about the calibration, standardization, and sampling. The first point is that calibration and standardization. Now, calibration is done on the regular basis to calibrate and standardize the equipments and instruments. Now, it is very necessary to calibrate and standardize every instrument before going to commit experiment. It is the process of determining the accuracy. Now, through this calibration, we determine the accuracy of the instrument. It involves the measuring the observation and comparing with them to standard. Now, standard results are available. We just compare with them for the standard. All measuring instruments, apparatus, and volumetric glasswares used in experiment needs to be calibrated to ensure they are offering accurate results. So, before going to start the experiment, it is needful to carry out the calibration with the help of good accuracy for instrument, apparatus, and volumetric glasswares. For example, balances can be calibrated using standard weights. Now, all the balances are analytical balances can be calibrated using standard weights. Now, these weights are universally accepted. Next one is standardization is the process. Now, this is all I have already learned. I have already said about the part of calibration. Now, standardization. The second paragraph. Standardization is the process of establishing the relationship between the amount of analyte and methods of observation and signal. So, standardization is nothing but it gives the balance between the amount of analyte and observations and signals. For the standardization of method, we use calibration of graph for the standard concentration of analyte with known concentration. The method's observation changes with respect to amount of analyte. So here, standardization is done with the help of calibration curve, or we can say linear curve method. So with the help of, like in colorimeter, we have already performed. Now the next one is sampling. Sampling is also a crucial part of the analysis. When we select a sample from the large portion and convert them into a small portion. Now this small portion is regarded as a laboratory sample, and from this laboratory sample we take some part for the analysis, and that analysis, the part of sample taken for the analysis is regarded as a analytical sample. So this is the main difference between the analytical sample, laboratory sample, and large sample. A sample is a portion of selected material from the larger quantity of material. The process of collection of samples is called as sampling. So sampling is nothing but the process of collection of sample is called as sampling. It represents the whole material. Now the sample represents the whole material. It is a representative of whole material. For example, 1 liter water is taken from the lake. Now for a lake contains a lot of reservoir of water. Now we take a small quantity, that is a 1 liter, and that 1 liter is regarded as a sample. Now from that 1 liter we take a 10 ml for the laboratory sample, and from that 10 ml we take a 1 ml for the analytical sample. So that is a major difference. This sample is called as a gross sample. The gross sample is further reduced to a small quantity called as a laboratory sample. From the laboratory sample, we select a very small sample called as analytical sample or analyte. Now the analytical sample is also called as analyte. Now this analyte contains sample as well as the portion which under consideration for the analysis. The analyte should be homogeneous and it must be similar or resemble the composition of total mass of the material. So the analyte is also the representative of whole material, or you can say total mass of the material. After collecting analyte sample, the sample treatment is given for drying and resolution of sample in suitable solvent, which acts as a stock solution. Now, after collecting a analyte sample, a processing is done, and that processing is done for the drying and dissolution of the material. Now, this dissolution of the material in a proper solvent, which is regarded as a stock solution for the analysis. So, this is all about the calibration, standardization, and sampling, which is the essential part of the development in procedure after once method is selected in analytical method or in analytical analysis. So with this content, I conclude this lecture. Thank you very much for your patience listening.